This video will cover how you will go about writing up the actual coursework for, try again. This video will cover the coursework for PY167, and this is the write-up entitled Investigation into the Effect of the Opioid Morphine on the Motility of the Guinea Pig Gastrointestinal System. It will cover the actual write-up itself, the abstract, the results, and the conclusion that you actually have to write. It will also cover how you go about collecting the data from your field-stimulated demonstration that you had during the pain case, as well as collecting the data from the colonic motility assay with the pellets that you also saw during the demonstration. In addition, it will also cover how you will use your data that you collected this week from your practical class. So, as a brief overview, what you will be writing is that you will be writing an abstract, which is a 200-word maximum piece paragraph. We will want you to indicate your word count in brackets at the end, but what I advise is that you actually leave the abstract until the end, so that you've got all the results written up and your discussion before you return to the abstract, which is going to be a summary of the actual experiment. The introduction is written for you, and we recommend that you read this. This will give you some idea about how you go about writing an introduction to an experiment, something that will stand you in good stead for coursework that you will encounter during the rest of your course. Additionally, we have also written the methods for you. And again, I suggest that you read the actual methods and see exactly how you go about describing how you do an experiment and how you do and how you would explain this in an actual report, so that somebody could, if they read this document, would be able to repeat what you did in the laboratory. The actual methods as well may give you some idea about how maybe you would like to present your work in terms of being logical and how it all fits together to give a nice story about the actual experiments that you've done and how you would go about discussing and describing what you found. What you do have to do is you have to complete a results section. And for the results section, you have to draw 5 graphs. And in a moment, we will switch on to the data, and I will indicate how you go about collecting the data for the molar concentration of morphine versus the contractile response of field-stimulated tissue graph, and the molar concentration morphine versus percent relaxation of stimulated tissue graph. In addition, we will look at the data for the colon motility and the fact that you want to graph that up as treatment versus velocity. And then we will come on to the last part from the data that you collected this week, where you worked and had it looked at the effects of acetylcholine and how you will graph up your molar concentration of acetylcholine versus contractile response in the unstimulated tissue, and also the log of the molar concentration of acetylcholine versus the contractile response in unstimulated tissue. We will also want 2 tables in your results section. One looking at the effect of naloxone on morphine atropine mediated effects on field stimulated tissue, and a second table, table 2, on the effects of morphine, naloxone, and atropine on acetylcholine mediated effects on unstimulated tissue. We will also require a commentary on your results alongside the figure legends. Finally, you will then write a discussion of 650 words, and we will come to that once we have dealt with the results section. We would also like you to reference your work, so we are looking for references in full, and as a result they are not included in your word count. And likewise, we're looking for an appendix, and we're looking for an appendix in terms of putting in all your calculations or your tables where you've done the calculations, so that if there is a problem with the graphs, we should be able to identify that and be able to provide feedback on that. We are looking for a word count for the entire report of about 1,100 words plus 10%. You can go up to 1,200, but any more than that, and that will be a problem. We will stop marking at that point. So if you have 1,300 words, the last 100 words will basically not be looked at. Your lab report is to be completed and submitted electronically by midnight on the 17th of March 2019. Do not leave it to the last minute. If it is late, even by 1 minute, then that will obviously mean that there will be repercussions for the marking of that report and for your overall mark. We have also put the assessment criteria here, and what I suggest is that you actually read this assessment criteria fully so that you know exactly what we are looking for when we are marking this document, but we'll return to this at the end. Okay, so the first thing we'll do is that we will look at the data from the field stimulated contraction, and we will have a look at how you go about collecting the data from that and what sort of graphs we want from that. Okay, so how do you get your data from the actual trace that you have? The thing you are looking at here are with the large arrows where you've added the drug. And in this case, it is morphine. These last two are where we've added naloxone, but we'll deal with those in a moment. So, the first thing you need to do is you need to get a response where there was no morphine present in the bath. So where are you going to get that response from? Well, you're going to get it from one of these 5 peaks right by here. So what you're going to do is after you've printed it out, you are going to measure one of these peaks. Once you have measured one of those peaks, you will get that in centimeters. And at that point, you need to convert that into grams. And the way you do that is by looking at this scale bar down here. That distance there is equal to 1 gram. So you measure your peak, you measure the scale bar, you divide your peak by your scale bar, and that will give you the number of grams that the response was. And that's what you do for all the morphine concentrations. So where do you measure for the morphine concentrations? Well, as I said, each of these arrows is where the morphine has been added, and it is increasing dosages. So this first arrow is 1 times 10 to the minus 8. This second arrow is 3 times 10 to the minus 8. This third arrow is 1 times 10 to the minus 7. This fourth arrow is 3 times 10 to the minus 7. And this fifth arrow is 1 times 10 to the minus 6. So for 1 times 10 to the minus 8, which of these bars are you going to actually measure? Well, considering that you added the drug here, the drug is going to have its most effects here, just before you add the next concentration. So it is this one, which you will measure for 1 times 10 to the minus 8 and convert to grams by dividing it by the scale bar. Then you need the distance for 3 times 10 to the minus 8, the response. Which one of these are you going to measure? And again, it's going to be this last one, because that is where the drug has had its maximal effect. Similarly, for 1 times 10 to the minus 7, it's going to be the last one by here. Similarly, for 3 times 10 to the minus 7, it's going to be this last one by here. And for 1 times 10 to the minus 6, it's going to be this last one by here. And that is what you're going to measure and convert into grams. As part of the data collection, and what you need for your results is that you need the effects of naloxone on, as you can obviously see, morphine-mediated inhibition of field-stimulated contraction. So this is where you've added 1 times 10 to the minus 5 molar naloxone, and this is where you've added 3 times 10 to the minus 5 molar naloxone. I know it's run off the end here, but it's basically 1 times 10 to the minus 5, 3 times 10 to the minus 5. I don't mind which one you use. You can even use both if you want to, but it doesn't particularly matter. So what you do is that you added naloxone there. So if you were going to measure that response, the effect of naloxone on the morphine response, you would measure that peak. If on the other hand you're going to use the naloxone 3 times 10 to the minus 5 molar, you obviously measure the last one by there. And it doesn't matter which it is. You only need one response to actually put into the actual table. So that's how you collect the data for the actual field-stimulated guinea pig ileum effects of morphine. The other slide that we've given you is the effects of atropine on field-stimulated guinea pig ileum contraction. Now, in terms of the data, we're not asking you for for you to draw up a dose response curve of the effects of atropine on field stimulated contraction. But what we are asking you to do is to put into the table the effects of naloxone on atropine-mediated inhibition of field-stimulated contraction. So what you're going to need to do is that you are going to need the response of atropine 1 times 10 to the minus 8 molar. So again, similarly to the last experiment with the morphine, you need to measure one of these peaks. Which one are you going to measure? The one just before you add the naloxone. And then at that point, you can decide whether you're going to use the 1 times 10 to the minus 6 molar naloxone or the 3 times 10 to the minus 6 molar naloxone concentration to put into your table. If you're using the 1 times 10 to the minus 6, you'd measure this peak. And if you're using the 3 times 10 to the minus 6 molar, you'd measure this peak. It doesn't matter what. And again, it's a similar process. You measure it and get it in centimeters. That is your scale bar. That equals 1 gram. So all you need to do is divide the size of the curve the size of the response that you've measured and divide it by the scale bar to get your response in grams. And of course, if you put all this lot into a word table, all this data can then be put into your appendix so that we can actually see what you've done. We can see what you've measured. We can see how you've converted it to grams. And as we come on into a moment, we'll see how you've converted it into milligrams. And at that point, we can determine if there are any errors or things like that. So that's how you collect the data for the field stimulated tissue, and how you get the data for basically the first 2 graphs that you have to draw and table 1. Okay, so once you've got all your data, once from the field stimulated tissue, what are you actually going to have? Well, let's deal with the morphine data first of all for the data that you actually require for a graph. And what you're going to have is you're going to have a table, and that table will have a column labeled concentration, column labeled centimeters, column labeled grams, and a column labeled millinewtons. And we'll deal with the last one in a second. So in terms of concentration, you are going to have 0, 1 times 10 to the minus 8, 3 times 10 to the minus 8, 1 times 10 to the minus 7, 3 times 10 to the minus 7, 1 times 10 to the minus 6. And that's morphine concentration, and that's going to be in molar. You will have done the measurements, so you may well have measured this, and you might find that that is maybe 40 centimeters, maybe that's 38 centimeters, maybe that is 24 centimeters, maybe 10, maybe 6, and maybe 2. And as I indicated on the actual slides, you'll have measured the scale bar, which equals 1 gram, and you will have divided the response in centimeters by whatever that scale bar might be measuring. So if it was measuring 1 centimeter, for example, 40 centimeters divided by 1 would give you 40 grams. 38 grams, 24, 10, 6, and 2. That's if the scale bar is 1 centimeter equals 1 gram. Once you've got it in grams, what we want you to graph up is actually the response in millinewtons. So how do you calculate that? Well, 1 gram equals 9.81 millinewtons. This is obviously not going to be an exact calculation, but you're going to multiply the 40 by 9.81, and that maybe might come to 380, something like that. So you just multiply up your response in grams by 9.81. So that might give you these sort of numbers. Again, as I said, these are very rough because of the fact that I'm not doing the calculation. Once you have the response in millinewtons against your concentration, you're now ready to draw up the first graph. And the first graph that we want is along the x-axis, morphine concentration in molar, and response on the y-axis in millinewtons. And when you draw that up, the graph should look like something like that. When you're drawing up the graph, we actually want to see on the graph the actual points marked in, either with a cross, with a square, with a triangle, it doesn't matter. But we want to be able to see all the points. And we do want to see a line joining those points up. You can try a line of best fit. Excel will allow you to do that, but it may not be perfect because of the way the data is. So I would suggest you just join up the points with a line. And that looks like your first graph, and that's it. It's done. The key thing to remember is about the concentration. These are not real numbers as such. If you type them in exactly like that into Excel, your graph will be incorrect. The reason being is because 1 times 10 to the minus 8, so you would have to type in an X in that middle bit. That's a letter. So Excel would recognize this then instead of not as a number, but as an actual word. So what you actually have to do is you have to put these in as real numbers, which means for 1 times 10 to the minus 8, for example, that's going to be 0, point, and then there are 7 zeros. 1, 2, 3, 4, 5, 6, 7 before the 1. Similarly, for 3 times 10 to the minus 8, it's going to be equal to 0, point, and again there is 7 zeros. 3, 4, 5, 6, 7, 3. Okay, and you have to do that for each of those. If you have those in as your concentrations, real numbers, and you put in the response in millinewtons and you draw the curve, that will give you the graph that we are looking for. And that should be no problem. So that's your first graph. The next graph that we actually want is percent relaxation. And I suppose actually in the actual table, you could actually put another extra column in to actually cover the percent relaxation. So if you insert a 5th column in as percent relaxation, you can do that calculation. So essentially, how do you do that? Well, here we've got 0 molar morphine. So what's your percent relaxation going to be when there is no morphine in the actual bath? Well, of course, it's going to be 0. So how do you actually calculate that? Well, it's going to be 100 minus your response for that concentration, which in this case is 380. It's going to be divided by the response of 0 morphine, which is also 380, multiplied by 100. If you do that calculation, 380 divided by 380 is obviously 1, multiplied by 100 will be equal to 100. 100 minus 100 is 0. And that's what essentially you will get. That's 0% relaxation. So your actual calculation for percent relaxation is equal to 100 minus your response divided by the response when there is 0 morphine. Multiplied by 100. So if we do this one here, which is 200, it's going to be equals 100 minus 200 divided by 380 multiplied by 100. And that's because the response is 200. The response with 0 morphine is 380. So that's where you get the 200 divided by 380. You multiply by 100. So it's not quite a 50% relaxation. So it may well be that that will be maybe a 45% relaxation. With 98, that is well above 50% relaxation. It might even be as much as 60% relaxation. So you might calculate that to be 60%. The 50, again, it's going to be maybe 75%, and maybe the 19 is going to be 92% relaxation. You will do the calculations for your data. And then you need to draw up the graph, and that's what we would expect. The second graph, again, is morphine in molar along the x-axis, but this time along the y-axis, it's going to be percent relaxation. And what this time you're going to get is you're going to get a curve which this time goes up. And again, we want to see the points marked on the curve, and we want to see those points joined up with a line. And just as with the previous graph, when it's the morphine concentration, you obviously need to convert the 1 times 10 to the minus 8, which is not a number. You remember it's 0 point, and there is 7 zeros. 1, 2, 3, 4, 5, 6, 7, 1. And you need to put that in as for the numbers. And that will get you your second graph. And so that's the data that we require, and it should be fairly straightforward. This table here, once you've drawn it up and it's in Word, etc., that table can go in your appendix. Your graphs will go into your actual report. Okay, so that is the first two graphs. The next thing we'll talk about is Table 1. Okay, Table 1. This again relates to field stimulated data. And this table, what we want is the effects of naloxone on morphine and atropine. You would again have collected that data in centimeters. So again, what we are looking for in the appendix at least is again a table before naloxone, after naloxone, that you would have subdivided this into centimeters, grams, millinewtons, centimeters, grams, millinewtons, and that in this column would be basic, the first column would be treatment. And so you would have morphine, and you'd have atropine. And again, you'd have the concentration underneath here. And I can't remember what it is from the actual data, from the actual data graph. I suspect it is 1 times 10 to the minus 6 molar. I can't remember what it is for atropine. You need to get that data. Make sure that the concentrations are marked in on the table. And the same with the naloxone concentrations. You may have to indicate that there are 2 different concentrations being used, in which case you can actually leave it as before naloxone and after naloxone and refer to that difference in concentrations for the different treatments in the actual table legend. I leave that choice up to you. And again, you would have done the measurements. So with morphine, it might be that it's 3 centimeters, which would turn out into 6 grams, which, if you're remembering that 1 gram equals 9.81 millinewtons, that turns into probably about 58 millinewtons. And after naloxone, that centimeters may have increased maybe to 25 centimeters. That in terms of grams would be 50 grams. And then you multiply that by 9.81 to get your response. So that's going to be something like 480 millinewtons, something like that. And so that's what you'd have in the table. And the same with atropine. With atropine, it might be 2 centimeters, which might turn into 12 grams, which may then have turned into 110 millinewtons. And then afterwards, you might find that it's maybe 3 centimeters, that would basically have turned into maybe 6 grams or something like that, and then in which case that may turn into 55 Maybe something like that. And then you would show the different effects. And you draw your conclusions from that. That is the table that would go into your appendix. But in your actual report, you would abbreviate that table. So that table then would only have treatment, before, and after. And again, it would have the treatments with morphine and atropine. Again, with the concentrations. And the before and after naloxone would all be in millinewtons. So you would just have one value there, one value there, one value there, and one value there. And that basically would be table 1. And that finishes the data collection and the data presentation for the field stimulated data. Before we move on to the color motility, let's talk about figure legends. So what you are going to have had is basically 2 figures, morphine, molar, against response in millinewtons, and that would be going something like that, remembering the crosses to mark the points and everything else. And your second graph would be, again, morphine in molar, but this time against percent relax, and this one would be going up. And what I would do is I would put those two together, because they're very often showing the same sort of thing. And I would call it A and B, and I would call that figure 1. If you look at graphs in research papers and things like that, you find that there are no titles. You don't need a title on the graphs, but that is what the figure legend is for. You'd have figure 1, and then you'd write about these figures. And what I usually tell people is that the first sentence of your figure legend basically equals the title. What is it? So that is basically a one-liner that you would say something like, effect of morphine on field-stimulated contraction of the guinea pig ileum. Full stop. That's it. Your second sentence of a legend, and it can sometimes be more than that, depending on how many figures you have. But in this case, you've only got one effect. So your second sentence describes what the figure shows. And in this case, it is morphine causes a dose-dependent inhibition of field-stimulated contraction. Full stop. And that's it. I won't be talking about the legends for the tables, for the colon motility, or for the unstimulated ACH tissue, because I've basically given you this one. Now you need to go and write the others for the tables and for the other graphs. Follow this example and it should be not a problem. But that's what we want you to do in terms of the actual legends. It is, as I said, first sentence title, second sentence what happened. The reason why we do this is because of the fact that ideally what you would like to be able to do is pick up a research paper and without reading anything, be able to look at the graphs and read the figure legends and know what is going on. To know what the actual graphs are showing and what the actual sort of conclusions that you can draw from that graph, what they actually are. Maybe not the underlying mechanisms or things like that, but you can get from the graph a sense of what the actual data is about and what the paper is about. And that's what we expect here. And so, as I said, it's very nice and clear. First sentence, what the actual graphs are showing, what the actual experiment is, the title in a sense. So that is the effects of morphine on field-stimulated contraction of guinea pig ileum. And then the second sentence with what happened. Well, if you look at the data, you can see that morphine dose-dependently inhibited field-stimulated contraction. So that's all you have to say. So copy that example, do that for the next graphs and the next tables. Okay, the next thing we're going to look at is the data from the colonic pellet motility experiments. Again, that was indicated in the demonstration. You saw it in the demonstration, and we provided you with some data here. So what's the key data that you need to take away from this? The first thing is that the length of the colon, which is 7 centimeters. Okay, because you're going to need that to be able to calculate velocity. In this first column, you have the treatment. You have 2 values for the control, 2 values for 10 micromolar morphine, and 2 values for naloxone in the presence of morphine. So this is where you've added the naloxone after you've added the morphine. So this is the effect of naloxone on the morphine. What we've given you is time taken for the pellet to travel 7 centimeters in seconds. And you have 44.5, 35.8 for the controls, no movement in 9 minutes, no movement in 9 minutes for the morphine, and then 19.4 and 19.6 for naloxone. So what we are looking for you to graph up here is a column graph. And what we would like is basically 3 columns. So how are you going to get that? Your first column is going to be control, the second column is going to be morphine, and the third column is going to be naloxone. So how do you turn 2 values into 1? Essentially, you could just pick one of the values, but better for science, really, is that if you took the mean time taken to travel 7 centimeters for the control, the mean time for the morphine is obviously with no movement, 0 and 0, so there's no movement. And then the naloxone effects on morphine is basically 19.4, 19.6, so you take the average of that and you draw that up. But please remember, this is not naloxone alone. This is naloxone added to the bath after you've added the morphine. So it's basically the effects of naloxone on morphine, and you can draw your conclusions from that. And what we would like you to graph up for this is, as I said, a column graph, but what we're looking for is these times, these effects of the control, morphine and naloxone on the pellet motility to be converted into velocity. So, for the actual graph for colon motility, what we are looking for is at the bottom you're going to have control, morphine, remember to put the concentration down, and naloxone plus morphine. And again, remember to put the concentrations. Okay, so your graphs all need to be fully indicated with concentrations, etc., so that we know what each of the treatments are, what it actually is. And on the y-axis, velocity, which is going to be centimeters per second. And you're going to draw this up. So you're going to have maybe a curve like that. That is obviously going to be 0, so it'll be down there somewhere. And then this will be a reversal, something like that. So you need to do the calculations. We've given you the time it takes to travel 7 centimeters. So what does velocity equal? Velocity equals distance over time. So distance is always going to be 7 because that's the 7 centimeter colon, and then you put your responses here and you calculate the velocity and you graph that up. And again, remember that we want, because this is going to be figure 2, some sort of legend. And that's what we expect you. So as I said, I'm going to let you come up with that. So that's all you need to do for the colon motility, for the data to draw up basically figure 2. Okay, so that brings us on to the final bit of data, the data that you collected. On the unstimulated guinea pig ileum. So, hopefully, what you collected was the data for the concentrations of ACH for the dose response curve. This is your part 2. And you remember that you had 1 times 10 to the minus 8 molar, 3 times 10 to the minus 8, 1 times 10 to the minus 7, 3 times 10 to the minus 7, 1 times 10 to the minus 6, and 3 times 10 to the minus 6 ACH molar concentration. And you have recorded a response, but this time it's in grams. So what you need to calculate from that is a response in millinewtons. And again, I'm expecting to see this sort of table in the appendix, because this is where you're going to do your calculations. So your response of 1 times 10 to the minus 8, it may well be 0, or it may be 1 gram or something like that. Your 3 times 10 to the minus 8 might be 2.4. Maybe your 1 times 10 to the minus 7 is 4, maybe 6, 12.8, or 12.6, something like that, and maybe okay, so finally we come on to the last bit of data, which you need to draw up for basically figure 3 and table 2. And that data is what you collected this week from the experiment, which is from the unstimulated Okay, this brings us to the last bit of data which you are going to need to draw up basically figure 3 and also table 2. And this is from the unstimulated tissue from the experiment that you did this week. And if you remember from the unstimulated tissue, the first graphs that we want, or the final 2 graphs that we want, are from the dose response curve to ACH. So you have ACH in molar, and this was your part 2. So you remember you've got 1 times 10 to the minus 8, 3 times 10 to the minus 8, 1 times 10 to the minus 7, 3 times 10 to the minus 7, 1 times 10 to the minus 6, 3 times 10 to the minus 6. And if you remember, what you actually have, or what you collected, is that you collected baseline and you collected peak. And we expect that to be in the actual in the table that's going to go into your appendix. And so you collected your baseline, and maybe your baseline was 0.2 for that one, maybe 0.3, 0.1, it could be minus 0.4, there it could have been 0.6, and there maybe it is 0.1. And then your peak might have been something like 0.4, maybe 0.6, maybe 0.9, maybe 1.1, maybe 1.8, and then maybe 2.4. And what need you to calculate then is the response. And obviously the baseline is in grams, the peak is in grams, so the response is going to be in grams. And the response equals peak minus the baseline. So when you do that calculation here, it's going to be peak 0.4 minus 0.2, so that's going to be 0.2, 0.6 minus 0.3 is 0.3, 0.9 minus 0.1 is 0.8. Here, remember, it is going to be 1.1, but it's minus the base, so minus and minus becomes plus, so that maybe becomes 1.5. Here it becomes 1.2, and here it's going to be 2.3. And what I'm highlighting here is that ideally you would hope, because as you increase the ACH concentration, that you would get an increase in response. But as you see in the example here, I've indicated that these two didn't actually do that. And that could have happened in your data. Okay? And that's okay. We're not marking you based on the data. We're not taking marks off if it's not doing the right sort of thing. This is biological data. It can be quite hard to get it to give you the perfect dose response curve. With the tissue, obviously there is peristalsis going on possibly. This is one experiment. It is one bit of tissue. Maybe the bubble stopped for a bit and it didn't have enough oxygen, or maybe there was a depletion of ATP to cause contraction. Any sort of things like that could have happened. So it's nice if you've got the perfect dose response curve where it's going up with all the concentrations. But if you don't, that's absolutely fine. And obviously then you need to convert the response to millinewtons. 1 gram equals 9.81 millinewtons. So you have to do the multiplication up. So as I said, 0.2 times by 9 is maybe going to be 2 millinewtons. That may be 2.4 millinewtons. That might be maybe 8.4 millinewtons. That might be 15 millinewtons. That might be 12 millinewtons. And that might be 20 millinewtons. And that's what you will get from your data. And from this data, now this is where we want you to draw up two graphs. And what those two graphs will be is ACH concentration on the x-axis in molar versus response in millinewtons. And what we're looking for is a graph, again, that will go up like that. And again, we need to see the points on the line. And we need to be able to have a line that joins up those particular points. I haven't marked it on here, but obviously in these experiments, 0 ACH will give you 0 response, will give you 0 peak, will give you 0 millinewtons. That's supposed to be millinewtons. I apologize for that. Okay, so that's what you're going to do the calculations for. And so there would be a 0, 0 here. And in your graph, you can put that in. But yeah, as I said, you draw up the graph like that. The second graph that we want is slightly different. The x-axis remains the same. No, y-axis rather remains the same, as in results in millinewtons. Your response in millinewtons. But this time the x-axis is going to be different. And the x-axis, we actually want you to log the molar concentration of ACH. Okay? Now, first thing to note is you can't log 0. So what you have to do is, once you've put all these numbers in to get this graph, is take away the 0. If you do that, it changes this graph. So your graph will start here. So you set the graph so it does not start at 0. Once you've done that, all you've got to do is double click on this axis and tell it that you want it to be a log. And when you log it, what you're looking for is what you expect maybe from a typical dose response curve. So we're looking for like an S-shaped curve. You may not get that. We may not have done enough concentrations to have enough at this end, which would be flat line, which would be 0, which would have very little effect, or to have enough at this end to actually get to your maximal effect. That might have happened, that's fine. But that's the sort of shape we're looking for. So there you're looking for the ACH molar concentration on the x-axis, and for the other graph you're looking for the log molar concentrations of ACH. The key thing about this is to take away the 0. Removing the 0, double click on the axis, and Excel will actually do this for you. You don't have to log it. You don't have to get the calculator out and log it. You don't have to do that. Excel will do this for you. Once you've drawn up those particular graphs, we obviously want a figure legend. And again, you can call it A and B, and you can put your figure legend in, and this is going to be figure 3. And you write what the figure legend is. And as I said, I've given you an example for the first one. This time you have to come up with your own. So it is fairly straightforward to do this because Excel will do the log for you. If you leave the 0 in, it will look very odd because the graphing just doesn't like doing that, so it would not look quite right. But apart from that, it should be fairly straightforward. As for the morphine graphs, please remember, these are not numbers. You must convert these back to the usual 0.1, 2, 3, 4, 5, 6, 7, 1 to get 1 times 10 to minus 8. You must convert them into numbers. Okay? If you don't do that, your graphs will look odd. Because, as I said, Excel thinks that these are actually words. So what it will do is it will equally distribute them along the x-axis. And at that point, what you will end up with is essentially a straight line. And when you do that, you will find feedback will be written on your report saying this is not a linear scale. And that's the key thing. It needs to be a linear scale. And the only way you can do that is by writing the numbers in. That should be fairly straightforward for the ACH response. And to get your figure 3 for the 2 graphs. The final thing to talk about the data is Table 2. Table 2 is again coming from the unstimulated data. And what we're looking for is the effect of morphine, naloxone, and atropine on ACH-mediated contraction. So the final table in your graph should look something like this. ACH before, ACH after. And remember, you must put in the concentrations because otherwise we won't know, we won't know what you've used. So be detailed. That's the key thing. Then down here under the treatments, you're going to have morphine, again with the concentration, whatever it may be. You're going to have naloxone, and you're going to have atropine. And again, these results here, before and after, are going to be in millinewtons. And it may well be that your morphine in millinewtons may be 10 millinewtons, and then afterwards it might be 11. With naloxone, it might be 8 millinewtons before and maybe 10 millinewtons afterwards. And for your atropine, it may be 12 millinewtons before, and it might be 2, it might be 0, it might be whatever you want. Some of you had to put in more than one concentration of atropine, so you should probably put them all in. So you can actually do that. Atropine, you can put the concentration, you can put another concentration of atropine and another concentration of atropine if you so desire, and to show the effects that you obtained. So that is the actual final table 2 that we're actually looking for. And as with the figures, obviously the tables have figure legends. You would put a figure legend in. What is the title? Well, I've sort of slightly given that away early on, but then what the actual effects are, and be able to do that in your second sentence. So what does that actually mean for, how do you actually get this data to be able to put into the table? So the data. What you've essentially got is you've got ACH before. You will obviously have treatment. And what you would have underneath here is, again, baseline, peak, response, and then response in millinewtons. And those would be grams, grams, grams. Similarly, you'll have ACH after. And again, you're going to have the baseline, the peak, the response in grams, and the response in millinewtons. And you will have that inside your appendix. So we've done that calculation. Your baseline might be 0.4, your peak might be 1.6, so your response is going to be 1.2. You're going to be searching millinewtons, so that would be about 11 millinewtons. And then, afterwards you might have 0.3, you have 1.4, so in which case you've got 1.1, and then your things might be 10.1 or something like that. And you'll have that for all the actual your treatments. And that's the table that you would have in your appendix. And again, we can see then whether you've done the calculations correctly when we look then at your table 2 if there are particularly any issues. And that's it. That's all the data collection that you actually need. What we're going to talk about next is the actual write-up itself. So we'll talk about the results section and we'll talk about the discussion. So results. We've talked about the graphs. So you are going to have 3 graphs, essentially, or 3 figures, I suppose. Okay, let's talk about the results section. You are essentially going to have 3 figures, 2 tables. I will leave you to decide the order of how you're going to assemble this. But what I will say is you can read the materials and methods and see the order that they're in. That might give you some ideas. And also the fact, I would say, be logical. Think about the story you're trying to tell. So what 3 figures are you going to have? You're going to have the morphine and field stimulated. And like I said, you're going to have A and B, and you're going to have your legend underneath. So that's going to be figure 1 with a legend. And that's going to be the morphine effects. You are also going to have table 1 from the field stimulated data, and that is going to have the effects of naloxone on atropine and morphine, effects on field stimulated contraction. You've got figure 2, or another figure 2, which is going to be your colon motility. And your figure 3 is again going to be possibly A and B. And this is going to be your ACH response with molar concentration of ACH versus response in millinewtons and response in millinewtons, and this time the log of the ACH concentration, log molar concentration. And you're going to have table 2, which is basically the effects of morphine, naloxone, and atropine on ACH-mediated contractions, what you actually got. And that's what your results section is going to contain in terms of the data. We need a legend on the figures and on the tables, and we need it to put into a logical order. What else do we need in your results section? Well, we need a results commentary. So what is a results commentary? Well, a results commentary is basically a description of what you saw. So how you divide it up is entirely up to you. What order you put in is entirely up to you. But for example, if you're looking at the field stimulated data, what is your results commentary? What happened? Well, you know that inhibited field stimulated contraction. Now, yes, you don't have any figures for this. You haven't drawn it up in a graph. We told you not to. But you can still put it in. And you could refer to that as data not shown. And that's absolutely fine. What else did you discover in the field stimulated contraction? What was the morphine effect? And so you state that in a sentence. And obviously the morphine effect is seen in figure 1. So you refer that to figure 1. What else did you look at? You looked at the effect of naloxone on morphine, on atropine. Table 1 rather. So what did you see? And so that's what we expect the commentary to be. A description of your results. No conclusions. Don't draw any conclusions. Don't try to put in mechanisms or anything like that. It is purely a commentary. Lots of people worry here because in this experiment, your commentary might suddenly look an awful lot like your figure legend. That's okay. So you have to consider the fact that this is a lab practical. We understand that this is a fairly straightforward practical. It may not have felt that way when you were actually doing it this last week, but it is actually a relatively straightforward practical, and the results you've got are fairly straightforward. So the commentary is going to be relatively simple. You can, of course, add stuff into your commentary if you so desire. You could maybe do a calculation for an atropine or 2 atropine concentrations and put that in as maybe percent relaxation. You could give an example of the morphine, the concentration that maybe caused close to 50% relaxation or something like that. So you can spice up your commentary if you so desire, but you don't need to. And so just a straight commentary describing your results is more than enough. That would be field stimulated. You obviously have colon motility commentary to write, and you have a commentary to write on the unstimulated tissue. With the word count, we count the words in the legend and in the commentary. We don't count the words on the graphs, so don't worry about doing that to that sort of degree. But we do count the words in your commentary and in the words in the legend, so you have to keep to the word count for those. Which is why I suggest keeping the commentary nice and simple. Also, this is a results section. So, what are you going to do? Are you going to write all the commentary and then put all the graphs? Does that look right? Are you going to write all the commentary, put all the graphs, and then put all the tables? Do you think that looks logical? Have a think about how you present this. Do you have to have all the graphs together? Could you interleave them with the text? Could you insert them into the text? I will leave it up to you for the presentation. There is presentation marks. We will be looking for good presentation. As I said, have a look online. Have a look at some of the published papers. They will give you an example of how the results section should be sent out, as well as giving you some ideas about what a commentary looks like. But as I said, in this case, it is very, very simple. Okay, the other section you need to write. Okay, the other discussion you need to write is the discussion. And this is 650 words. So what am I going to tell you about the discussion? Well, ideally, not very much. We want you to actually write the discussion. We need you to write this and to, put together what the experiment actually is. This is quite hard to do because of the fact that the practical, we know what the effects are of morphine, of atropine. We know what they do and we know how they work. You can just open up Rang and Dale and it's all in there. But what we want you to try to do, at least in the first paragraph or the first section of your discussion, is to pretend that you don't. So draw your discussion. Discuss your data. What does your data show? So if I gave you one example here, it's the sort of thing that we're looking for. Field stimulated. It was inhibited by atropine. What does that mean? What's the link between the two? So what is this? What does atropine do? You know what it does. It inhibits ACH. So if it inhibits ACH and it also inhibits field stimulated contraction, what does that mean? What can you conclude from that? Well, you can conclude from that field stimulation, the actual contraction, is mediated by ACH. What does that mean? Well, as we said, field stimulation is where we are applying electricity, an electrical current across the tissue. So the electrical current could directly contract muscles. What else can it do? Well, if it was directly contracting muscles, would atropine have an effect? Think about it. Would it? No, it wouldn't, because obviously the electrical impulses will be independent of atropine. So that means the electrical effect is in having an indirect mechanism of causing contraction. And that indirect mechanism is mediated by ACH. Where does the ACH come from? Think about the indigestion case. What have I been talking about? ACH is a neurotransmitter in what system? In the enteric nervous system. So, from your data, from looking at the data that you've got, you can make these conclusions. But field stimulated contraction is not being mediated directly by electricity having a direct effect on the muscle contraction. But it's an indirect effect, but it's through ACH. And the ACH, because that is located in the enteric nervous system, that therefore the electrical stimulus must be stimulating the enteric nervous system to release ACH, which is then causing a contraction. That's the sort of discussion, that's the sort of thing that we want you to put together. And yes, it's quite easy for this particular practical because we know what the effects are. But when you come to the fourth year, when you are doing your research project, we won't know any of this. In your research project, you're doing stuff which is new, which has never been done before. So in terms of those experiments, then you have to sit down, you have to work through those experiments and think and draw conclusions from it and draw the underlying mechanisms of that. Field stimulation. What's the effect of morphine? What did it do to field stimulation? It inhibited it. How? What conclusions can you draw in a mechanism for morphine? What could morphine be doing to affect field stimulated contraction? Could be, maybe, relaxing the tissue. If you relax the tissue, you make it harder to contract. What else could it be doing? Well, ACH. You know ACH is responsible, so maybe it's inhibiting the ACH receptor. Could be. Or three, what else could it be doing? Could be affecting ACH release. Inhibiting it. There are three possible mechanisms. Which one is it? And how do you know which it is? And answering Rang and Dale tells me so is not really what we're looking for here. Because you've done the unstimulated tissue. So if you've done the unstimulated tissue, what does that say about this? What does it tell you about the mechanisms? What was the effect of morphine on the unstimulated tissue? Why does that give you an idea about what the mechanism is in the field stimulated contraction? And that's what we're looking for you to discuss in the first section. Obviously, you've got some naloxone data as well to fit in to talk about as well. to try to discuss why are we putting naloxone in? Why are we showing you naloxone? Think about it. Naloxone reverses morphine but doesn't reverse atropine. What does that mean? Have a think about it. Draw that sort of discussion. That's the first section that we're looking for. And I don't know how many words that will take. But also in the discussion, we want science. We want detailed science. If you look at the marking scheme, there are marks for the conclusions, for the discussion, for scientific definitions, for the added material. So in the second section of your discussion, we want to know why. Why are these effects happening? What's the underlying cellular effects? ACH. Why is it causing contraction? We're thinking of receptor. We're thinking of second messenger systems. And we're thinking about how the second messenger systems cause contraction. We're now looking for the scientific detail. Morphine. Again, why does it have its effect? How is it having its effects? Receptors, second messenger systems, etc. What the effects are on the cell. We're looking for the scientific detail. We're looking for, why these things are happening in specifics. Okay, just general stuff will get you some marks, but we want absolute specifics in this. That would be the second section. And the last thing I would say is, in terms of a discussion, how do you end it? Do you just tail off? Do you just stop mid-sentence? You need some sort of conclusion. What haven't I mentioned in the discussion? What experiment haven't I mentioned? What would be good for the conclusion? Clinical relevance. Can we make it clinically relevant? Morphine. Side effects, maybe. Morphine is an opioid. What do we use opioid? What can we use opioids for? Which experiment didn't I mention? Could you put that into a clinical context and relate it maybe to opioids, maybe to morphine, maybe related to side effects, maybe related to clinical relevance? Could any of the data that we have talked about here support perhaps therapeutic approaches or anything like that? So again, have a think about this. We need you to think about this. We need you to think about what these experiments mean, what it all means, and then put that into the actual write-up. And that should be it for the discussion. No problems. References. We want them in full in the section. Okay, because they are not part of the word count. So word count, not important for the references. Not counted for the reference section. You know, we've talked about reliable, or you should have talked about reliable references. Is Wikipedia a good source? Yeah, I'll let you think about that. What other sources do we have? What would we like you using? Obviously, we'd like you to use journal articles. These experiments were done by a guy called Patton back in the 1950s. You could find the original report. Could be interesting to read, perhaps. Journal articles, obviously, textbooks. Websites, think about, are they reliable? Perhaps if it's a charity, yes. WebMD, less so. So, think about what your reliability of the sources are to make sure we use the most appropriate references. How are you going to reference in the text? Numbered. Okay, it needs to be numbered in the text because obviously we're going to count the words. If you don't, in the discussion, if you put in the full references and it's not numbered, that will add on to your word count. So there is the Journal of Pharmacy and Pharmacy Practice, which we have recommended that style before. That is a numbered style. When you are referencing, so in your discussion, say if you tail off in your sentences something like, morphine activates X receptor, and then you need to reference that. So what you normally do is you would superscript, and maybe the reference number is number 1, maybe number 2. And what you would do is that you would superscript it, no spaces. Do not put a space between receptor and that first bracket. Do not put a space between the comma and the 2. Do not put a space around the brackets at all. If you keep all of that together, it will count it as one word, which means you have written a sentence and referenced it, and you have not used up any words. If you try to put this in as, for example, if you're referencing maybe Mabley et al., 2002, 1, 2, 3, 4 words you'll use up out of your word count. You put in like 10 references and that's 40 words gone. So you must use numbered. It would help you with your word count. And like I said, do not put spaces with a bracket. Just superscript it. Put number 1, number 2, both numbers, one number, whatever it might be, 3 numbers altogether. It doesn't matter. And if you do that, it won't add into the word count. So that's the reference section. And again, you should be able to score pretty good marks on the reference section. It's worth 5%. Put them in full. The other thing about reference section, consistency. Make them all look the same. Okay, if you're using just the first author and then et al., that's what you do for all of them. If you're using 2 authors then et al., that's what you do for all of them. The order that you put it in. Title, the year before the title or the title before the year, it doesn't matter. Make sure that all the page numbers are there, the journal article, journal volumes are there. Do not have some authors in block capitals, others not in block capitals. All of that will lose you marks. Be consistent in your approach and you will score better marks. Appendix. It's worth 5%. So make sure you put all the stuff in, the data tables, where you've done all your calculations. Okay? Put them all in. That's all you have to do. If you put all the data tables in for all of the stuff that you've done for the field stimulated, for the colon motility, for the unstimulated tissue, all those data tables are in, then that's it. You'll get the full 5%. So it's worth putting in. You get 5% even before you start. Even without us reading it, if you've put all that in, you immediately start on 5%. So make sure you remember to put it in. If you leave out the appendix, you obviously will get 0 for it. If you don't reference your work, we can't give you any marks for it, so we'll obviously get 0. So don't leave any sections out. Okay, I said we come back to it at the end. Abstract. Have a read of research papers and see how they write the abstract. How would I describe the abstract? I basically describe the abstract as almost like a movie trailer. It's designed to make you want to read the paper. Does that mean that everything has to be in the abstract? Well, no. you won't be able to put everything in the abstract. You've got a lot of data here to talk about. You've got a lot of data. So your abstract, what you need in your abstract is background, aim, methods, results, and conclusions in 200 words. It's not easy. I would suggest you could use these as subheadings inside your abstract. Lots of journals do that. But your background has to be basically one sentence, maybe two. So what's your background going to be? Think about it. What's the title of your experiment? What's it referring to? What are you looking at? And using that, you should be able to devise a background sentence. Linking to the background, you basically want one sentence for your aim. And again, what's it going to be? What is the experiment's goal? Link it to the background. Methods, keep it brief. You don't need full methods in here. Okay, so you've seen the methods section that we've written. So you've got to just abbreviate that into basically, if you can get down to one sentence, that would be great, but probably 2 sentences max is all you can do in terms of the methods. These are the key sections here, the results and the conclusions. So results here, you can allow yourself 3 sentences, maybe. But go with the key results. You don't have to go with them all. Don't have to explain them all. But you can put in the key results that tell a story, that tell the story that after you've talked in the discussion about, that's what you can refer back to and think about. So when you've written your discussion, you know the story that you're telling. So in which case you can use that to devise which are your key results and put those into your abstract. And then you need a conclusion. And again, I try to keep the conclusion to 1 sentence. What conclusion can you draw from this? What are you aiming? Okay, you may go for 2 sentences for your conclusions, but you've got to keep it quite tight. And again, as I said, you don't have to talk about every single bit of data that you've got. All you actually have to do is just, is talk about the key story and what the key conclusion is. What's the key conclusion from your work? You might be able to make it 2 sentences, possibly, and again, put maybe a bit of a clinical aspect on that last sentence, on why your work, why this experiment is important, why your work is important. So something like that. It's not easy writing an abstract, but as I said, it's not worth a huge amount in terms of marks. Most of the marks in this report are around your discussion and your results section, and in your discussion, a lot of marks in the discussion for how you discuss the data and how you put the scientific stuff in. And that's because you've never written anything quite like this before, not for therapeutics, in a therapeutics theme, in a therapeutics experiment. So that's why that's set up like that. So take a shot at it and write the abstract. Once you've done that, you've finished. Submit the work. Remember to put your name on remember to indicate the name of the experiment and everything else. Don't put your name on it. Obviously, we're now working off numbers only. So just put your number at the top of your work. So on the off chance that something gets uploaded wrongly or something goes wrong, we can actually tell your piece of work because your student number will be on there. But please leave your name off it because we're doing it anonymous these days. If you have any questions, then obviously you can email me. Please make sure you get this in by the deadline. The last thing to look at is the assessment criteria. It is always good to read this. It tells you what we're looking for. The title, you don't have to worry about it. Okay, I need to alter this one. It says also remember to add your name, student ID, practical lab group. All we need is your student ID and your practical lab group. We do not need your name on this, okay? Because we are doing anonymous marking, so I apologize for that. So ignore that little bit there. The abstract, we just talked about it, 200 words. And as it says there, it should summarize the study. And a good idea is to split this into introduction, methods, results, and conclusion. And I say 2 sentences per section, but if you can get down to less than that, it gives you more room for other things. It should be the last thing you write. As I said, if you can write it at the end, you know what your abstract is supposed to be summarizing because you've done all the discussion and all the results. So that's why I say leave it to the end. It's what we do when we write papers. 200 words, it's worth 10% of the overall mark. As we've indicated, we've given you the introduction. It's worth reading it, and you're probably worth reading it based on the fact that you will have to have some idea of the introduction because you have to write your abstract. Similarly with the materials and methods, we've written this, but it would be good to read it because obviously you have to refer to the methods in the abstract. So you've seen the methods, you've done the methods in some cases, but you obviously need to, you have the full methods there to be able to go into the abstract. Then there's your results, and we've talked about that. So the results is 250 words overall, and it's worth 20% of your actual report. We're accounting for the fact that, things like using Excel is quite hard and things like that. So getting used to drawing up figures, drawing up graphs, and analyzing your data, we realize that you may not have done that so much, which is why it's 250 words, and it's why it's only worth 20%. You must have a commentary, otherwise you will lose those marks. And you must have the graphs. If you leave any of these graphs out, we obviously can't mark them and you will get 0 for that particular section or that particular part of the results section. So we're looking for the molar concentration of morphine versus contractile response of field stimulated tissue in millinewtons. And we're looking for the molar concentration of morphine versus percent relaxation of the stimulated tissue. We've talked about this. The third graph is treatment versus velocity in centimeters per second for the pellet motility assay. You also then need the molar concentration of acetylcholine versus contractile response in unstimulated tissue, and the log of the molar concentration of acetylcholine versus contractile response in unstimulated tissue, and you got that from the experiment this week. As I said, I would expect you to combine graphs 1 and 2 into a figure, A and B. That would be figure 1. Graph 3 to be into a separate figure, which would be figure 2. And expect graphs 4 and 5 would be into figure 3. And then you label them A and B. You obviously need the 2 tables. You want the 2 tables of the effects of naloxone on morphine and atropine on the field stimulated tissue, and the effects of morphine, atropine, and naloxone on ACH in the unstimulated tissue. And you need table 1 and table 2. Again, you will need figure legends and table legends. No title is needed on the actual graph. The first sentence of your legend is the title. We've talked about this. That should be fine. For this practical, statistical analysis is not required because you've only, for the most part, done n equals 1, and even for the colon motility, it is only n equals 2, and you're not going to be able to get reasonable stats out of that sort of data. So you don't need statistical analysis for this practical. Then we're talking about the discussion. Start your discussion with a couple of sentences summarizing your main findings. We've talked about this. You need to integrate the experiments together to actually discuss your observations and results, and draw conclusions from that. And then obviously then talk about the underlying scientific concept of those, which we'll come on to into a second. But also then using references to existing literature. So you do need to have references in here. We are looking for you to describe the effects of field stimulation and acetylcholine on ileum contraction, and then using the data collected from both the field stimulated and acetylcholine-induced contractions to explain the cellular mechanisms and the discussions and the conclusions that you can draw on the actions of morphine, naloxone, and atropine. And at the end, make sure you draw some conclusions. So you need references in this. It's going to be 650 words, and that's worth 40% of the mark for this lab report. The scientific content is really what we're looking for as well. That's worth 20%. So in terms of your discussion part is worth 40, then this on top is worth 20. And this is, most of your scientific content is going to be in the actual discussion. Although, of course, there is using the appropriate units on graph axes. There's obviously the bits in the commentary and using appropriate terminology in the actual legends and things like that. You know, inhibited, for example, is scientific. Decreased is maybe not, and things like that. So being specific and using the scientific appropriate terminology will get you the marks here. We will look at whether you directly related the pharmacological effects correctly to the actual effects that we're seeing. As I said, we need more than just a receptor name. Naming drugs as an agonist and antagonist, we need more details than that. We require, receptors, second messengers, cellular effects, things like that. And that's worth 20%. And we've talked about that already. Referencing and citations, we've talked about. It's 5%. The actual reference section is not included in the word count. The actual numbered bit is. Okay, so whatever words you put into the discussion, we will count those. Okay? There is no way around that, because obviously all we can do is highlight it. If we had to separate out all the individual words of cited references in a discussion, then in which case, we couldn't do that very easily. So you must remember to use the numbered version. Lecture notes, I would probably not use as a citation source, because it's better to go with textbooks, reviews, journal articles. And as I said, web-based sites, okay, but make sure you analyze and determine whether you think that the actual information you're getting from those sites is real, is relevant, and is true. How reliable are those sites? Your reference section is worth 5%. No references, you'll lose that mark. So make sure that you actually have a reference section. And same with the appendix. We want to see all the raw data tables. Don't necessarily need to see what we've given you in terms of those slides or anything like that. What we're looking for is mainly the data tables. The stuff where you've done the measurements, you've turned it into grams, and you've turned it into millinewtons, and things like that. When you calculated the velocity and that sort of thing. So put all of that in there. Here it says Power Lab output. As I said, you don't need that here. We're looking for mainly the calculation tables. You don't refer to the material inside the results of a discussion in the appendix. The appendix is just there that we can look at to see whether or not you've done the calculations correctly. And again, it's worth 5%. So if you don't have an appendix in there, in which case you will lose those marks. There is, under the assessment tab, more detailed guidelines on how to write a successful lab report. You've already written up some reports and some other coursework associated with previous modules. Read that feedback. Have a look at the feedback for that and determine how you can improve for this particular report. And you should be fine. As I've said before, when is the actual deadline? If you flip back here, I'll repeat it again. Deadline submitted electronically by midnight on the 17th of March 2019. And again, I will also say, if you have any questions or queries, then please email me. I'm sort of happy to help you. I'm obviously not going to give you answers. I'm not going to tell you about receptors and second messages and so much stuff like that. But if there are problems maybe with things like Excel or how you do something in Excel or things like that or anything, then obviously email me and I'm likely to respond with an answer. Thank you.